All right, let's crack open the toolbox of genetic engineering. Today, we're looking at something that's absolutely crucial, but maybe a bit of an unsung hero, the cloning vector. You might not have heard of them, but these things, they basically powered a revolution in modern genetics. So let's dive in and see how they actually work.
So think about this for a second. A single gene. It's like one sentence in a library filled with millions and millions of books. It's an invisibly small piece of information. How in the world do you find it, get it out, and then make a ton of copies so you can actually study it? I mean, that's the massive problem that early molecular biologists were up against. So how'd they do it?
Well, they solved it with this absolutely brilliant concept, the cloning vector. The easiest way to think about it is like this. It's a delivery vehicle. You can't just drop a letter in the mail without an envelope, right? It's the same idea here. You can't just shove a gene into a cell and hope for the best. You need something to carry it. And that simple idea, that analogy of a delivery truck for DNA, is going to be the key to understanding everything we talk about next. So where did scientists find these first delivery vehicles? Well, they found them hiding in plain sight, right inside common bacteria. And this brings us to the OG, the original workhorse of genetic engineering, the plasmid. Okay, so plasmids are these fascinating little things. They're these small, circular pieces of DNA that bacteria have, completely separate from their main chromosome. And a lot of the time, they carry handy genes, like for antibiotic resistance. The reason they were so perfect for scientists is that they're small, they're simple, and most importantly, they copy themselves. This made them the perfect candidates for the first cloning vectors. Think of them as a reliable pickup trucks of the genetic world, perfect for carrying small loads of DNA.
But of course, scientists didn't just use them as is. They engineered these natural plasmids to make them even better tools. They basically gave them a toolkit with three essential parts. First, you've got the origin of replication, or ORI. That's the ignition switch. It tells the host cell, hey, start copying me. Then there's the selectable marker. This is like a license plate. It lets you identify which cells actually have the plasmid. And finally, you have the multiple cloning site, or MCS. This is just a specific, engineered spot where you can easily cut the plasmid open and paste in your genetic cargo. Super clever.
Okay, but there's a catch. Getting bacteria to actually slurp up these plasmids from their environment, it's a process called transformation, and frankly, it's not very efficient. At all. You might get only 1 in 1,000 or even 1 in 10,000 cells to actually take up your plasmid. So how do you find that one successful cell in a massive ocean of failures?
And that is where that license plate, the selectable marker, becomes an absolute game changer. Remember how I said it's often an antibiotic resistance gene? Well, you just take all your bacteria and grow them on a petri dish that's full of that antibiotic. And what happens? Only the bacteria that successfully grabbed your plasmid, the one with the resistance gene, can survive and multiply. All the others, they just die off. It's such an elegant way to filter out all the failures and find exactly what you're looking for.
So, plasmids are amazing. They really are. But just like our pickup truck analogy, they have their limits. They're great for small jobs, but their cargo capacity is pretty small, maxing out around 15 to 20,000 base pairs. If you try to stuff more in, the whole system just gets unstable. So that raises the next big question. What if you need to move a much bigger piece of DNA? Well, if you need to move a bigger load, you need a bigger truck, right? To solve this problem, scientists looked to nature again, and they found the ultimate delivery experts: viruses. I mean, when you really think about it, what is a virus? It's basically a prepackaged biological syringe perfectly designed to inject its own genetic material into a host cell. So scientists had this aha moment. They decided to co-opt one of these natural experts, a virus called bacteriophage lambda, which loves to infect E. coli, and basically hijack its incredibly efficient delivery system to carry their own, much larger genetic cargo. And the way they did it is so clever. See, the lambda virus DNA has this big chunk in the middle called the stuffer region that it doesn't actually need to infect the cell. So, scientists use molecular scissors, enzymes, to snip out that non-essential stuffer part. Then, they paste in their big piece of foreign DNA right in its place. Finally, this newly modified DNA gets packaged back into the virus particle, turning it into a custom delivery vehicle. And this isn't just theory, this is the real deal. What you're looking at here is an actual map of one of these early phage vectors. You can literally see that central stuffer fragment that gets taken out and replaced. By doing this, they were able to create vectors that could carry up to about 24,000 base pairs. Now that is a serious upgrade from our little plasmid pickup truck. But you know how science is, right? The ambition just keeps growing. A semi-truck is great, but what if you need to move something even bigger?
Soon, researchers were staring down the final frontier, how to clone and study truly enormous pieces of DNA. And for that, they needed something completely different. I mean, what if you want to study an entire biological pathway with a whole network of genes that all work together? Or maybe a huge piece of a human chromosome, something with hundreds of thousands of base pairs. Yeah, for a job that big, a pickup truck isn't going to cut it. A semi-truck won't either. You need a true cargo ship. And this is where our story takes a really cool turn. We're moving away from just cleverly modifying things found in nature and into the realm of pure invention. Scientists realized that to carry a load this massive, they couldn't just hijack another natural vehicle. Nope. They were going to have to build one from the ground up. They were going to build an artificial chromosome. So if you're going to build a chromosome from scratch, what's on the parts list? Turns out you really only need 3 critical components. You need a centromere, which is kind of the handle that the cell uses to pull chromosomes apart when it divides. You need telomeres, which are like the protective plastic caps on the end of shoelaces to keep the ends from fraying. And, just like our plasmid, you need an origin of replication so the cell knows to make copies of it. Armed with this blueprint, researchers in the 80s went to work, building the first little artificial chromosomes in yeast cells. They put all the right parts together, a centromere, telomeres, and origin of replication, and it was a complete and utter failure. These little artificial chromosomes were ridiculously unstable. The cells would just lose them almost immediately. They had all the right pieces, so what on earth was going wrong? And then came the big breakthrough. The key insight was that the problem wasn't what parts they used, but the overall size of the thing they built. Researchers discovered, almost by accident, that if they made the artificial chromosome long enough, we're talking over 50,000 base pairs, all of a sudden it became stable, like really stable. It just started behaving like a proper natural chromosome. It turns out for chromosomes, size really, really matters. And the difference wasn't small, it was absolutely staggering. Just look at this chart. It shows the mitotic loss frequency, which is just a fancy way of saying how often the cell messes up and loses the chromosome when it divides. The short little guy at 11 kb was lost all the time, but the longer 55 kb artificial chromosome, whoa, its loss rate just plummeted. It was almost 20 times more stable. This was it. This was the key that unlocked the ability to clone massive, massive pieces of DNA. So let's just take a step back. We've gone from these little pickup trucks, the plasmids, to semi-trucks with viruses, all the way to building our own massive cargo ships from scratch. So why does this whole journey, this evolution of tools, matter so much in the real world? Well, this table really sums up the entire fleet of tools we've built. You've got your plasmids, the pickup trucks for small jobs, the phage lambda vectors, our semi-trucks for medium-sized loads, and then you have the real heavy hitters, the bacterial artificial chromosomes, or BACs, which are our cargo ships carrying up to 300,000 base pairs. And the biggest of them all, the yeast artificial chromosomes, or YACs. These are the super tankers of the fleet, capable of carrying over 1 million base pairs of DNA. And look, this fleet of vectors isn't just some neat scientific curiosity. These tools are the invisible workhorses, the engines that powered the entire genomics revolution. Think about it. As the cargo capacity of our vectors grew, so did our scientific ambition. We went from studying single genes with plasmids to mapping entire genomes with BACs and YACs. Let me be clear. The Human Genome Project, it would have been completely impossible without these tools to carry and copy the massive chunks of DNA needed to piece it all together. So this whole journey, from finding a tiny plasmid in a bacterium to literally building a synthetic chromosome from scratch, it represents a fundamental shift in our power to engineer biology. We're not just borrowing and modifying nature's delivery vehicles anymore, we are building our own. And that leaves us with a pretty incredible question to think about. Now that we can do this, what will we engineer next?